As the prevalence of antibiotic resistance continues to increase at an alarming rate, driven in part by the lack of new antibiotics being developed, researchers around the globe are working to identify potential antibiotic candidates to help combat this global crisis. And now, one such candidate has been found by an unexpected source, artificial intelligence. And how this discovery came about and what this means for AI's potential role in combating the antibiotic resistance crisis will be the focus of today's discussion. Welcome to the Clinicians Roundtable on ReachMD. I'm Dr. Paul Doghramji, and joining me today is Johnathon Stokes, a postdoctorate fellow at the Broad Institute of MIT at Harvard. He's also the lead author of the study that identified this powerful new antibiotic compound. Thanks so much for joining us today, Johnathon.
Hello.
So before we jump right into the study, I'd love to learn more about how you started exploring the use of artificial intelligence to identify new antibiotic candidates. What sparked this line of research for you, Johnathon?
Yeah, so it was kind of interesting how it happened. Professor Collins and Professor Barzilai are the two corresponding authors on this manuscript, and they are faculty co-leads at J-Clinic at MIT. And J-Clinic is an effort to leverage AI approaches in healthcare. So being faculty co-leads, they both come from quite different academic backgrounds as well. Professor Collins is a bioengineer, and Professor Barzilai is an expert in natural language processing. When they started working together as faculty co-leads, they wanted to work on a collaborative project, and given our lab's interest in antibiotics and her lab's relatively recent interest in applying natural language processing approaches to chemistry, it seemed like an interesting collaboration to potentially use both of our expertise towards the discovery of new antibacterial molecules.
Well, that's very interesting background. Thanks for that information. So let's dive into some of the details here. What approach did your team take to reveal this new antibiotic compound?
For AI and machine learning models in general, you have to provide them with training data, examples of something that you want to see so they can make predictions on new things. So what we did is we took one of Professor Barzilai's lab's models that they have used previously for molecular property prediction as it relates to physicochemical properties, so things like solubility. So we took that general framework, and instead of asking the model to predict physicochemical properties, we asked it to predict biological properties. And in our case, this was antibacterial activity. In the Collins lab, we have a collection of about 2,500 small molecules. These include FDA-approved drugs, so we have 1,700 or so of those. And then we also have a collection of about 800 natural product compounds that are purified, which have been isolated from a variety of microbial and plant sources. So we have this library of 2,500 molecules. So at the start of the project, we screened those 2,500 molecules for those that inhibited the growth of E. coli, just in a 96-well plate. So then we had this growth inhibition data of these 2,500 molecules. And then we showed the model the structure of every molecule, as well as its corresponding growth inhibition activity. And that was binarized. So we have a molecule, did it inhibit growth, yes or no? So we have 2,500 molecule activity pairs. That's what we used to train the model on. And then with that model, we were able to run predictions for antibacterial activity specifically across different chemical libraries, both housed here at the Broad and the virtual chemical library that is the Zinc 15 database.
Okay, and the results, what exactly did you find?
After we trained our model on the 2,500 molecules, we first applied it to the drug repurposing hub housed at the Broad Institute. And the drug repurposing hub contains both drugs that are used in the clinic for, a variety of diseases, as well as molecules in phase 1, 2, and 3 clinical trials, and those that are in preclinical development. So when we applied our model to the drug repurposing hub, we were looking for molecules that were both highly likely to be antibacterial, meaning that they had a high prediction score. The model strongly predicted that they were antibacterial. But the other constraint that we put, we only were interested in molecules that were structurally dissimilar from known clinical antibiotics. And that is measured in something called a Tanimoto score. So Tanimoto similarity is a way of comparing two different structures of antibiotics and saying how similar are they. So when we were looking for molecules, we wanted molecules that had a low Tanimoto similarity to any clinical antibiotic. And the molecule that fit those two criteria, right, structurally dissimilar from known clinical antibiotics and strongly predicted to be antibacterial, was this molecule that we ended up naming halicin.
Very interesting. So now, as I understand it, several other potential antibiotic candidates were also found using this machine learning algorithm, and they show potential for new drugs based off of their chemical structures. So what components make up these particular molecules, and what makes them so much more powerful than current antibiotics?
Right. So after we had ran our predictions on the drug repurposing hub, discovered halicin, we then leveraged our model to predict molecules from the Zinc 15 database. And the Zinc 15 database is a virtual chemical library that's composed of about 1.5 billion molecules. So instead of applying our model to all 1.5 billion molecules, we selected a subset of around 107 million on which to run predictions. And just for reference, in a wet lab, your upper limit of molecules that can reasonably be screened is on the orders of a few million. You're not going to find very many screens that are beyond that. So us being able to analyze the structures of 107 million molecules puts us at 2 orders of magnitude greater than that, which we thought was quite exciting. And anyway, from these 107 million molecules, again, we prioritized those that were highly likely to be antibacterial, as well as structurally dissimilar from known antibiotics. So with those constraints, we were able to curate 23 of such molecules for testing in the laboratory, and 8 of those 23 displayed antibacterial activity in at least one of the tested species. And we tested these molecules against just lab strain E. coli, MRSA, Klebsiella pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. Of these 8 molecules, we were particularly interested in 2 of these 8 because they displayed quite broad spectrum activity.
So for those just tuning in, this is the Clinicians Roundtable on ReachMD. I'm Dr. Paul Doghramji, and today I'm speaking with Johnathon Stokes from the Broad Institute of MIT and Harvard about the recent discovery of a powerful new antibiotic compound that was identified using this artificial intelligence. So Johnathon, now that we've covered what the study entailed and what was found, let's talk about its potential impacts. And based on the results, how do you think this may affect the global crisis around antibiotic resistance and its costly measures?
Yeah, so halicin's a really interesting molecule. It has a number of biological properties that we found quite exciting. So first, it's bactericidal, meaning it kills bacteria, but it's not only bactericidal against metabolically active and growing bacterial cells. It displays bactericidal activity against metabolically dormant cells. What's interesting is the vast majority of our currently used bactericidal antibiotics, they display great efficacy against metabolically active cells, those that are, active, growing, and dividing, let's say, but they fail to eradicate cells that are metabolically dormant. However, we showed that halicin was able to retain this bactericidal activity against dormant cells. And there's quite a lot of recent evidence showing that these metabolically dormant antibiotic tolerant cells, they're called, are responsible for chronic and recurrent infections in humans. So the fact that halicin was able to eradicate these cells irrespective of their metabolic state suggests that it may have the capability of doing this in vivo as well, which would prevent chronic and recurrent infections. The second thing that we tested was its ability to overcome various antibiotic resistance determinants, right? So we tested, a small collection of antibiotic resistance determinants in E. coli and showed that halicin was able to retain activity in the presence of different antibiotic resistance genes. And we also tested halicin against panels of carbapenem-resistant Enterobacteriaceae, Acinetobacter baumannii, and Pseudomonas aeruginosa. And we observed that halicin retained activity in the vast majority of CRE isolates we tested and Acinetobacter isolates we tested. We observed that it did not display strong efficacy against Pseudomonas aeruginosa. We don't have empirical evidence for this, but getting molecules into Pseudomonas remains a challenge. So we think it's just a permeability issue that we're dealing with. And the last phenotype that we observed is the frequency of resistance to halicin in the laboratory appears to be very low. So we ran an experiment. It was a 30-day long experiment where we tried to evolve spontaneous resistance to halicin in the lab using E. coli. So we had halicin, and then we compared it to how easily E. coli could evolve resistance to ciprofloxacin. And we observed E. coli began to evolve resistance to ciprofloxacin anywhere between 1 to 3 days. However, over the 30-day experiment, that we performed, we were unable to evolve resistance to halicin at all. And that's exciting because if the evolutionary barrier to evolving resistance is very, very high, as it appears to be, it may suggest that a molecule like this could have a prolonged shelf life in the clinic.
Very interesting way of inventing a new medication. So how do you think this might impact the future of biotech and pharmaceutical industries for new antibiotics?
Yes, I feel like the adoption of machine learning approach is beginning to permeate all aspects of our life, right? You know, we all get a different Amazon page when we open it up based on our previous buying history, all this stuff. So I feel like it's natural that these approaches will strongly influence the future of biotech and pharma, not only toward the discovery of new antibiotics, which we desperately need, but I feel like these approaches will also be leveraged, to find new cancer drugs or hypertension medication. I'm very confident, actually, that these approaches will become more widespread in therapeutics discovery and development in general.
Well, in line with that and looking at the future, what's next on the horizon for you and your colleagues? Will there be another line of investigations extending from this study sometime down the road?
Yeah, so the project that we're working on right now is leveraging very similar ML approaches to discover narrow spectrum antibiotics. So, most antibiotics are fairly indiscriminate. You take them and they'll treat, hopefully, your infection, but they'll also kill a lot of your microbiome, which then leaves you open to opportunistic infections like C. difficile. However, with a narrow spectrum agent, in theory, you'd be able to take such an antibiotic, it would eradicate your infection, but spare your microbiome. So that's the project that I am immediately working on right now.
That seems like a very nice way of approaching infectious diseases and in treating specific infections. Well, I certainly hope to have the chance to speak with you again, Johnathon, once those future developments are underway. But for now, I want to thank you for joining me and shedding light on how artificial intelligence is being used to usher in new antibiotics. It was great having you on the program today.
Thank you very much for having me.
I'm Dr. Paul Doghramji, and you've been listening to the Clinicians Roundtable on ReachMD. To access this episode and others in the series, visit reachmd.com/cliniciansroundtable, where you can be part of the knowledge. Thanks for listening.